This is the first of three videos on biochemistry. So we start off with some basics about protons, neutrons, and electrons that we need to be familiar with. Atoms are made of extremely tiny particles called protons, neutrons, and electrons. Protons and neutrons are in the center of the atom, making up the nucleus. Electrons surround the nucleus. Protons have a positive charge. Electrons have a negative charge. The charge on the proton and electron are exactly the same size, but opposite. Neutrons have no charge. Since opposite charges attract, protons and electrons attract each other. If the atomic number is 20, there are 20 protons. In a neutrally charged atom, there will be the same number of protons and electrons, so there are 20 electrons in this case. If the atom is neutrally charged, this means there is no charge. Protons are positive, electrons are negative. This is why they must be the same in number. Okay, so those are some basics that you might already be familiar with. Ions. If a neutral atom gains an electron, it becomes negative. This kind of ion is called an anion. If a neutral atom loses an electron, it becomes positive. This kind of ion is called a cation. Any atom with a charge is referred to as an ion. Amino acids are the building block of proteins. There are 20 different amino acids in the human body. We can synthesize 12 of them. We ingest the other 8. The key elements of an amino acid are carbon, hydrogen, oxygen, and nitrogen, although other elements are found in the side chains of certain amino acids. Enzymes accelerate chemical reactions. The molecules upon which enzymes may act are called substrates, and the enzyme converts the substrates into different molecules known as products. Almost all metabolic processes in the cell need enzyme catalysis in order to occur at rates fast enough to sustain life. Most enzymes are proteins, although a few are catalytic RNA molecules. The latter are called ribozymes. Enzyme specificity comes from their unique three-dimensional structures. Like all catalysts, enzymes increase the reaction rate by lowering its activation energy. Some enzymes can make their conversion of substrate to product occur many millions of times faster. And we have to look at some bonding. So first we look at covalent bonds. A covalent bond is a chemical link between two atoms or ions where the electron pairs are shared between them. A covalent bond may also be termed a molecular bond. Covalent bonds form between two nonmetal atoms with identical or relatively close electronegativity values. The electron pairs that participate in a covalent bond are called bonding pairs, or sometimes called shared pairs. Typically, sharing bonding pairs allows each atom to achieve a stable outer electron shell, similar to that seen in noble gas atoms. If two atoms each share an electron equally, the bond is covalent. Many organic compounds, such as carbohydrates, result from covalent bonding. There is a covalent bond between the oxygen and each hydrogen in a water molecule, H2O. Each of the covalent bonds contain 2 electrons, one from a hydrogen atom and one from the oxygen atom. Both atoms share the electrons. A hydrogen molecule, H2, consists of 2 hydrogen atoms joined by a covalent bond. Each hydrogen atom needs 2 electrons to achieve a stable outer electron shell. The pair of electrons is attracted to the positive charge of both atomic nuclei, holding the molecule together. Two important types of covalent bonds are are nonpolar or pure covalent bonds and polar covalent bonds. Nonpolar bonds occur when atoms equally share electron pairs. Since only identical atoms, which means they have the same electronegativity as each other, truly engage in equal sharing, the definition is expanded to include covalent bonding between any atoms with an electronegativity difference less than 0.4. Examples of molecules with nonpolar bonds are hydrogen, nitrogen, and methane. A water molecule, H2O, is an example of a polar covalent bond. The electrons are unequally shared, with the oxygen atom spending more time with the electrons than the hydrogen atoms. Since electrons spend more time with the oxygen atom, it carries a partial negative charge. A nonpolar bond, on the other hand, involves electrons being shared equally with a molecule. And lastly, we have ionic bonding. Ionic bonding is the complete transfer of valence electrons between atoms. It is a type of chemical bond that generates two oppositely charged ions. In ionic bonds, the metal loses electrons to become a positively charged cation, whereas the nonmetal accepts those electrons to become a negatively charged anion. Ionic bonds require an electron donor, often a metal, and an electron acceptor, a nonmetal. Ionic bonding is observed because metals have few electrons in their outermost orbitals. By losing those electrons, these metals can achieve noble gas configuration and satisfy the octet rule. Similarly, nonmetals that have close to 8 electrons in their valence shells tend to readily accept electrons to achieve noble gas configuration. In ionic bonding, more than one electron can be donated or received to satisfy the octet rule. The changes of the anion and cation correspond to the number of electrons donated or received. In ionic bonds, the net charge of the compound must be zero. You can recognize ionic compounds because they consist of a metal bonded to a nonmetal. Ionic bonds form between two atoms that have different electronegativity values. Because the ability to attract electrons is so different between the atoms, it's like one atom donates its electron to the other atom in the chemical bond. Ionic bonds are interactions between charged atoms or molecules. The highly polarized, which means charged, nature of ionic molecules is reflected in their high melting point.